Hey guys, welcome to Pre-AP Biology into our new unit. This is Unit 3, and we're going to be talking about cellular energetics. Yeah. Okay, so the first unit we're starting on, the first part, is going to be photosynthesis. And you should have your notes out for photosynthesis part 1, 3.1. Okay, and we're basically going to talk today about how the sun's energy is converted into a form that we can use. The first thing I want you to look at is look at this particular diagram. Okay, on this particular diagram, what we see is both matter and energy movement through the biosphere. And as you can see, energy is in red and chemical cycling, which is basically matter, is in blue. What do you notice about this diagram? One, you should notice that the blue arrows all go back into a circle. Its matter is being cycled. The amount of matter in the universe is pretty much constant, and the amount of matter on this planet is pretty much constant. We do not produce new matter. We just rearrange molecules. So your body is made up of molecules, atoms, and carbon and nitrogen and phosphorus that were once part of other organisms. It's just been recycled. If you look at the red arrows, however, you see that there really is no cycling of energy. It flows through ecosystems from the sun through the different levels of organisms, primary producers, primary consumers, secondary consumers, tertiary consumers, and so on and so on. And if you notice that at each one of those energy levels, heat is being given off. That's the wavy red arrow there to the on the moving towards the right in each one of these cases. All of the matter, however, and even some of the energy goes down to what becomes detritus. That just means like when an animal dies or when it poops and things like that, that material, the body, the poop and whatnot, can all be consumed by other types of organisms. And they are going to recycle that matter, but they're going to give off their energy in the form of heat. Okay, so that's an important concept. Matter is recycled, energy flows one way through the ecosystem. So those primary producers are extremely important, and those are the autotrophs. Auto means self, troph means feeding. These are the self feeders. Normally, we think of these as being plants. However, there are also algae. Algae grow mostly in marine ecosystems, but we also find it in freshwater systems and other damp areas. Algae is actually a group of organisms we call the Protista, and they can be large algae, such as a picture of this kelp, which can grow to be hundreds of feet long, to small microscopic algae, single-celled organisms basically. We also have phytoplankton, and phyto means plant, and plankton means a small unicellular organism. These are always found in both marine and freshwater ecosystems. These are single-celled eukaryotic organisms that undergo photosynthesis, such as this euglena, which is a single-celled organism. Many bacteria are also autotrophic. Okay, they're able to take in CO2 from the environment and then using the sun's energy convert that into sugars. And bacteria, the autotrophic bacteria, are what basically made this made this planet habitable or set the stages in terms of evolution for the evolution of multicellular organisms because they created oxygen. There was no oxygen on this planet until the process of photosynthesis evolved in the autotrophic bacteria, and they produced all the oxygen. We, however, as animals, are heterotrophs. We must get our carbon sources from other organic carbon sources, whether we're eating plants, such as this orangutan right here, or we are eating other animals. If you like to have a hamburger or a ham sandwich or something of that nature, you are a heterotroph eating another animal. Other heterotrophs are fungi. Okay, the fungi do not undergo photosynthesis. They have to break down other organisms, dead and dying organisms, and decompose those in order to get their carbon sources. They do not get their energy from the sun. They get it from the complex macromolecules, the carbohydrates, the proteins, and fats that are found in dead and dying organisms. Okay, so let's look at plants and let's look at basically how photosynthesis occurs in plants. The first thing we have to have is we have to have a structure in which photosynthesis can occur. And in the typical plant, that's going to occur in the leaves. And in the leaves, we find the organelle where this does occur, and that is the chloroplast. Okay, the chloroplast, if we remember back, was once a free living prokaryotic organism that became engulfed by a larger eukaryotic cell, and through the process of endosymbiosis, and we produced basically the autotrophic organisms came from that process. And in the chloroplast, we have to look at their structure. And you can see that it is a double membrane structure. The inner membrane, called the thylakoid, that's where we're going to find the molecule that actually traps and absorbs the energy of the photon, which is light energy, and that molecule is called chlorophyll. Okay, so the reason why plants are green is because the chlorophyll molecule is going to absorb all of the wavelengths of light except for green. Green gets reflected back, and that's why we see plants as being green. So if I was going to build a greenhouse and put some artificial lighting in there, the color of light that I would not choose would be green, because it would not be absorbed by the chlorophyll molecules, and therefore the plants wouldn't get any energy because they get their energy from light. But if they get it from green, since they do not absorb green wavelength, if I had a green light, they would not get any energy. All right, so let's look a little bit more closely at the chloroplast structure. As we can see, it is a double membrane structure. The inner membrane is highly folded. What's that mean? More folding, more surface area, more room for all the good stuff. Okay? And that highly folded inner membrane is called the thylakoid membrane. It is surrounded by a fluid called the stroma. Stacks of those thylakoid are called grana. Grana, and if I cut one of those discs in half, that would be the thylakoid space inside of that. And that'll become important when we talk about the production of ATP. So today we're going to talk about photosynthesis and the light reaction. Okay, as you can see on this particular diagram, we have the light reaction, which is taking place where? In the chloroplast, but specifically where? It's taking place on the thylakoid membrane, that inner membrane. That's where the light reactions take place. And as you can see, what are the inputs? Well, the inputs are going to be light and water. And what's the outputs? The outputs are going to be oxygen, ATP, adenosine triphosphate, the molecule of energy that all living organisms utilize to power their cellular processes, and another molecule which you have not come across yet, that's called NADPH. And what does NADPH do? It carries high energy electrons. Okay, where do the electrons get that energy from? They get it from light. And what's NADPH going to be used for? It's going to be used in order to basically reduce carbon dioxide in the next major phase of photosynthesis, known as the light independent reaction, or the Calvin cycle. And we'll get into some details of that on our next lecture. So there's the Calvin cycle, okay? And as you can see, the two Two of the products, ATP and NADPH, from the light reactions are going to be used in the light-independent reaction, or otherwise known as the Calvin cycle, and sometimes you may even might hear it referred to as the dark cycle or dark reaction. Okay, it doesn't need light. However, the Calvin cycle requires the products of the light-dependent reactions, ATP and NADPH, in order to do what it does. And what does the Calvin cycle do? It takes in carbon dioxide from the atmosphere. It's going to add some of those high-energy electrons, which are being brought into the Calvin cycle by NADPH. And when it adds those high-energy electrons to carbon dioxide, it's going to make it negative, and then protons, the H pluses, are going to want to jump onto that negative carbon dioxide and turn it into a sugar, CH2O. Okay, and because that is an anabolic dehydration synthesis reaction, it's going to require energy. Where's the energy coming from? It's coming from ATP. All right, so let's look at this chemical reaction. Okay, and remember, the conservation of matter states that matter cannot be created nor destroyed, so I have to have the same amount of atoms on the left side of the equation as I do on the right hand side of the equation. That's what we call a balanced chemical reaction. And look at the colors here. The colors represent where these molecules are going. And as you notice, on my reactant side, the left-hand side, we have simple molecules. We have carbon dioxide and we have water. We have 6 each of those. And the photosynthesis is going to produce sugars and oxygen and heat. Well, this is a building reaction. That means it's an anabolic reaction, so that means it uses energy. Of course, the energy, of course, is coming from the light from the sun or from your greenhouse lamp or whatever light source that you do have. All right, so look here. The carbon, of course, as you can see on my glucose side, that's that C6H12O6, is coming from the CO2 from the atmosphere. The hydrogens are coming from the water. And also look, the oxygen is coming from the water. So the air that we breathe started off as water. And then during the process of photosynthesis, we're using light energy to split water into hydrogen protons and electrons and oxygen molecules. All right, so let's look at the electromagnetic spectrum. Okay, what is the electromagnetic spectrum? It's basically the range of wavelengths that are being emitted. Most of these are all being emitted from the sun. We have gamma rays, X-rays, UV light, visible light, which as you can see is a very narrow range of the electromagnetic spectrum. Microwaves and radio waves. Now we use microwaves, we use radio waves, we use X-rays. We use these things for our own purposes, but the sun also emits all these also. Okay, but as you can see from visible light, okay, the electromagnetic spectrum ranges from 380 nanometers to 750 nanometers. A nanometer is a billionth of a meter. So these are very short wavelengths. Okay, and what's interesting, look at the bottom of there, the short wavelengths have more energy, the longer wavelengths have less energy. This is known as an inverse relationship. Okay, and light energy is a high quality energy. Okay, and what we can see is that the short wavelengths, the blues and the indigos and stuff like that, they have the higher energy amounts because they have the shorter wavelengths, and the red light has the lower energy amounts. Okay, now all these light waves together is white light. We blend them all together, but we can take a prism and we can divide these out into the different wavelengths. and we all know. So blue light has a lot of energy, red light has little energy, green light has a fair amount of energy, but plants reflect green light so they can't use it. And you may be familiar with Roy G. Biv. That stands for red, orange, yellow, green, blue, indigo, violet. Roy G. Biv. That's an easy way to remember how they go. Just remember that the violet is the shortest wavelength with the most energy, and the red is the longest wavelength with the least amount of energy. Okay, so this slide is showing how chlorophyll does absorb blue light and it does reflect green. So what we have here is we have a light, we have a prism set up that's going to split the white light into the different wavelengths, and we have a filter that's sending the green wavelength through this solution that contains chlorophyll, and then we have a photoelectric tube that's going to detect the amount of light. So if the light goes all the way through the tube, it's not being absorbed. And as you can see in this chlorophyll solution, green light goes right through the tube, and we're getting 100% or almost close to 100% transmittance, which means that none of that green light has been absorbed. However, if I do the same thing and I use a filter that only lets blue light go through, we can see that most of that blue light is being absorbed by the chlorophyll and very little is being passed through to be detected by the photoelectric tube, and we have very little transmittance. So blue light is a high quality light for photosynthesis. Green light is the low quality light for photosynthesis. Okay, and again, remember, in the thylakoid membrane, red, yellow, blue, and indigo and violet are being absorbed, but green light's being reflected, so that's why we see plants as being green. These couple of diagrams are important ones to know. The top one is absorbance of light by different types of pigments. Chlorophyll is a pigment. A pigment absorbs light. And all plants have chlorophyll A. And if you look at chlorophyll A, which is the blue line, okay, chlorophyll A absorbs light very highly in the indigo range, does not absorb in the blue to green range at all, nor in the yellow, and then it begins to absorb once again in the red range. Chlorophyll B absorbs a little bit longer wavelengths, more in the blue, and doesn't absorb green at all, and then absorbs a little bit again in the orange wavelengths. And the carotenoids, what sounds like a carotenoid? A carrot does. These are the yellows and the oranges. Okay, so if they're yellow and orange pigments, they're going to be reflecting the yellows. Okay, they do absorb in the blues and the violets and stuff. They do not absorb at all in the reds. Most plants have all three of these pigments plus some more. Bacteria usually only have chlorophyll A or chlorophyll B. And so, again, these are the different pigments and the different ranges of light at which they are absorbed. Now let's look at the middle diagram. The middle diagram is the absorption spectra. This is the action spectrum. This is looking at the rate of photosynthesis. That's what it says, rate of photosynthesis measured by the amount of oxygen released. And as you can see in the violet to blue range, we see a kind of an evening out of this curve, because again, this plant is using three different pigments, and so we're going to get an average of all those absorption spectra. In the greens, very little photosynthesis is occurring, and then as we go to the yellows and the oranges and the reds, we see some more photosynthesis occurring. Now on the bottom diagram, we see an actual experiment that was done using some algae, some photosynthetic algae, and some bacteria that are aerobic. What does aerobic mean? If you ever had to do aerobic exercise, that means you have to get a lot of oxygen into your body. Aerobic bacteria require oxygen in order to survive. And Thomas Engelmann did this experiment back in the late 1800s, where he took a filament of algae and then he put some aerobic bacteria onto a microscope slide, and then he took a prism and split light up to shine on top of this filament of algae. And what he noticed when he looked at the bacteria is the bacteria were clustered around the blue and violet range, very few in the green range, and a lot more back in the orange and red range. And if you look at Engelmann's experiment with the action spectrum, they pretty much are exactly the same. Why? Because photosynthesis was occurring at high rates down on the blue and violet range and also up at the red range. And what's a byproduct of photosynthesis? Oxygen. And what do these aerobic bacteria need? Oxygen. Okay, so they're clustered around the areas where photosynthesis is most active, and he was able to figure out from this simple experiment that these ranges of light, the wavelengths of light, the blue and the violets and the reds and stuff, were the ones that were most photosynthetically active. Okay, so let's look at the molecule where everything's happening. This is chlorophyll. Okay, chlorophyll is a pretty complex molecule that is found embedded in the thylakoid membrane. And if you look at the picture up here, the thylakoid membrane looks like what? It looks like a phospholipid bilayer. Okay, so the top part of the chlorophyll molecule is hydrophilic, meaning it loves water, and it sits on top of that membrane, whereas this long tail coming off the bottom is hydrophobic, and it's kind of like an anchor inside of that cell membrane. Okay, and at the center of that chlorophyll molecule is magnesium. And this is really important because if you look at it at a periodic table, you will see that magnesium is in column 2, which means it has how many valence electrons? They get boosted up to a higher energy level. And so now what do we got to do? Okay, let's look at this diagram. This diagram is known as the Z diagram. I expect you to be able to draw this diagram from memory. Okay, and I'll show you. It doesn't have to be as complicated as this. It can be a really simple diagram, but you have to be able to show the essential components. So let's look at this. What is happening? All right, so I just said that light is coming in, and it's coming in at photosystem 2 and at photosystem 1. But let's look at photosystem 2 first on the left-hand side. Light energy is coming in. It's going to excite those electrons. Those electrons get boosted to a higher energy level. And if you look on the left side of this diagram, we see the energy of electrons is increasing from the bottom to the top. Those two electrons, represented by 2e minus, are getting boosted up to a higher energy level. That's the 2 valence electrons from magnesium. Those electrons are going to get passed on to a protein that's called the primary electron acceptor, and all of this is happening on the thylakoid membrane. All these proteins are in the thylakoid membrane. All right, those high energy electrons go to that primary acceptor, and then you're going to get passed from one group of proteins to a next. This cytochrome complex is a specialized group of proteins that is going to pump protons against a concentration gradient. So if it's going from low to high, that is active transport. And active transport, if you remember, requires energy. Where is the energy coming from? It's coming from those electrons. Where do the electrons get the energy from? They got it from light. And as they pass down through this electron transport chain, what's happening to the energy of the electrons? It is decreasing. Okay, and those low energy electrons will finally go into photosystem 1, where once again light is going to come in and boost those up to a higher energy level, and they're going to get passed down a second but shorter electron transport chain. And there's a molecule there called, or I should say an enzyme called NADP plus reductase. What the heck is that? Okay, all NADP plus reductase does is it takes the substrate molecule, which is NADP plus, and it takes the 2 excited electrons and it hooks them up. It basically adds the 2 electrons to NADP plus, and that's going to, 1 electron will neutralize that and make it just NADP. The second electron will make this negative and will convert that, and that will cause a proton to join to it to make NADPH. Okay? And so that's where the electrons are going. That's called non-cyclic electron flow. So now let's all of a sudden let's go back to photosystem 2. We had those 2 electrons in the magnesium valence shell. They're now gone. They're now over here in NADPH. We now have an electron hole right here in the center of this chlorophyll molecule. What has to happen? We have to fill that hole. How do we fill that hole? We split water. Light energy is going to split water. It's going to form 2 protons. It's going to form 2 electrons. And that's where our oxygen comes from. Those 2 electrons from water are going to fill that hole.